I am honored to be here and speak to you today. And in full disclosure, I need to tell you what I shared with research colleagues at the Mitchell Cancer Institute yesterday. When I flew here, I had time to reflect on what I might say to you. And it occurred to me that all of you know an awful lot about oncology. And in fact, it reminded me of a true story. About 35, actually more than 35 years ago, I was on the national staff of the American Cancer Society. I was living in Chicago, flying out of O'Hare Airport, consulting on cancer prevention and control. All right, so I was flying around the country, and it was a typical morning, about 7 o'clock in the morning. I'm seated in United's boarding area, and seated next to me was a very well-dressed, dapper gentleman, black-vested pinstripe suit, graying hair, about 55 years old. Now, 55 is a lot older to me over 35 years ago than it is today. But a 747 airplane rolled by, and the man looked at me and said, "Boy, those sure are big airplanes, aren't they?" Little did he know that my true passion is aviation. And I said, "Yes, that's a Boeing 747 commercial airline, and in United's configuration, it seats 391 people." And he looked at me and said, "That is so interesting." So I gave him the entire dissertation on the 747. And I was concluding by telling him that on board that airplane is a computerized flight director. 100 feet above the runway, the pilot dials the flight directors in on the glide slope, punches the onboard computer, and doesn't touch the controls until the plane's on the ground. And the man looked at me and he said, "That is fascinating. You know so much about aviation. What do you do?" And I said with just that tinge of youthful arrogance, "I'm a national expert in cancer control research." "And what do you do, sir?" And he said, "I'm a pilot for United Airlines." So would you please take everything I say from here on in the context of that story? So I don't have any disclosures, but I do want to talk about the importance of population-based cancer research. And I'm going to do that in the context of the definitions of internal and external validity. And I want to talk about how population-based cancer surveillance programs can actually elevate our science in cancer research, how these concepts are related to the idea of translational research. Then I'm going to talk a little bit about our own cancer center's model for moving evidence-based cancer findings into the population. Then I'm going to give you an example of what happens when we do this. First of all, internal and external validity. I'm assuming all of you know this, so I am actually telling this to myself. I forget it and need to be reminded. In animal studies, if we use animals from POPs, we have essentially genetically the same animal. We can expose one set and not another, follow them over time to see if the outcome, and this can be developing disease or whatever outcome you want, occurs in the exposed animals versus the non-exposed animals. Because we control all of the factors in that experiment, we can draw causal conclusions and say that the exposure either does or does not contribute to the outcome of interest. Randomization allows us to do the same thing in human populations. If we have an adequate sample, we can randomly allocate a group of individuals to an exposure or a non-exposure. Oftentimes this is how we decide which treatments are best. But it's not just clinical treatments. We do exactly the same process in determining whether cancer prevention and control interventions are more or less effective. These kinds of studies where we control all of the known factors except the exposure are said to have very strong internal validity, and from them we can draw causal conclusions. So, while you're enjoying your lunch, I have a question for you. Randomization has been argued to be the most important scientific discovery of the 20th century. I agree with that. So why is it that the findings from our randomized clinical trials almost never work out the same in the general population as they did in the trial? Any guesses? Well, the answer to that question is the people in a clinical trial don't look like the people in the population. They tend to be much more well-educated, have more financial resources, better access to health care, probably a better diet, exercise more, and so on. So one of the things that we want to do is we want to look for external validity. How do the findings from our studies generalize to some underlying population? Epidemiology and population science provide us the tools to do that. And these are highly associated with particularly the last phase of translational research. I want to talk for just a second about 3 specific ways that population-based central cancer registries can actually elevate the science in our cancer research efforts. Population-based cancer registries gather information on every single cancer case that occurs in a defined geography. That means that we can provide investigators with either samples or complete enumerations of patients representing the underlying population. So if we use random samples or the full population, we get studies with strong external validity. This is just an example of a study that one of my colleagues, Dr. Ann Coker, and I were funded by NCI to do, and we were looking at whether intimate partner violence, violence against women in this particular case, affected the stage at diagnosis, treatment, and survival of women with breast, colorectal, and cervical cancer in Kentucky. We used the surveillance system and were able to get a scientific population of over 1,800 women. These were randomly selected, and they represent the underlying population of all of those cases. We found that about 37% of that population had experienced intimate partner violence at some time in their life. What's interesting about this study is that it appears that it does not matter whether that violence was as a child or more recent as an adult. It still had a great influence on the stage at diagnosis for those patients. Second reason that population-based surveillance systems can be valuable is that idea of rapid case ascertainment. Now, I'm going to acknowledge that we may be the only surveillance system that can fully do this right now in the US, but as I tell my colleagues, we're only about 2 weeks ahead of everybody else, so we better take advantage of it now. And the idea is that we as a surveillance system have incorporated natural language processing programs in all of the histopathology labs that will see tissue from Kentucky cancer patients. There are 48 labs across the state that's 400 miles long, 200 miles wide, both in and out of the state. There are actually 4 reference labs out of state that will see tissue. And these natural language processing programs just sit in the lab computer, and they read every path report. They determine whether it's cancer. If they decide it's cancer, they convert it to HL7 and immediately send it to our surveillance program. We estimate that we get 95% of all the cancer cases at the time they're diagnosed. So, and essentially we're getting it in real time. The importance of this is that we can identify very early on populations, and again, it's the population-based samples of patients who have very short median survivals, such as lung cancer or pancreatic cancer. And we can actually provide that information. We can consent the patients, get the consented patients to the investigators in time that they can actually collect tissue samples. One example is a large DoD-funded study that we're doing in Kentucky. The top panel here shows where there are high concentrations of arsenic. And arsenic and chromium are known carcinogens for lung cancer. And that tends to be in the eastern Appalachian area of the state. The bottom panel is another map of the state. The red area is the area defined as Appalachia, and it has extraordinarily high lung cancer rates, averaging in that 110 per 100,000 population. Some of the geographies go up to 130 per 100,000 population. Now, what we know is that certainly smoking is a culprit, but it does not explain all of these rates. So one of the ideas of the study is to see if it's not just the smoking, but the smoking in combination with an exposure to something like arsenic and chromium. If you know about lung cancer, you know that If you smoke, you have about 11 to 14 times the risk of getting lung cancer as a non-smoker. But if you smoke and you're exposed to, say, asbestos, that can go to 300 times the risk of a non-smoker. So we think that something like that may be going on. That study allowed us to get to those patients in time to gather additional information about their work history, their residential history, their smoking history, their diet history. We gathered blood, hair, urine, toenails. We wanted to have a pedicure service. No, just kidding. Radon levels, tap water, soil, and GPS. And this allows us to actually explore the role that these carcinogens play in the high lung cancer rates. So the last issue is the idea of thinking about these population-based surveillance systems not just as data, but as virtual. And again, the key word here is population-based because we want that external validity, population-based tissue repositories. And, oh gosh, it's been a decade ago we figured out we can do full genome deep sequencing on formalin-fixed paraffin-embedded tissue. And that changed the way, at least in our population in Kentucky, that we think about the surveillance system. So the population-based registry can serve as the honest broker for a study. They have statutory rights in almost every state to the identities of the patients. That means if an investigator wanted to look at tissue for a particular type of cancer, we know where those labs are. The registry can go and negotiate with them, collect the tissue, bring it back, anonymize it, and then it can be taken to usually a research lab for the processing or the PCRs or whatever you need to do. And it results in a study that has strong external validity as well as internal validity. An example of that is a study that I'm principal investigator on looking at the variations in HPV genotypes among women diagnosed with pre-invasive cervical disease. Back in 1996, somewhere in there, we all stopped collecting information on pre-invasive cervical disease because the definitions were all over the place and it was not possible to get a solid denominator. With funding from the Centers for Disease Control and Prevention, we did quite a bit of extensive research in 2008 and discovered that we could consistently define CIN3. The old marked dysplasia plus carcinoma in situ of the cervix. So that was our definition. We also could consistently get the adenoma the AIS cases, and those are a little bit different because they come from the lower stromal level tissue, but this is our definition of pre-invasive disease for the study. And it allowed us to ask some questions, and I'll go through those in just a minute. But we used that rapid case, we used the electronic pathology reporting, the natural language processing, to have all those cases reported to us. We went back, audited the labs. We could get roughly 100% sensitivity, about 92% specificity. So we were getting all the cases of these pre-invasive cervical disease with this definition. And so we're now addressing a couple of questions in this HPV genotyping study. The women who live in the Appalachian area of Kentucky are almost exclusively Caucasian. There are very few African American women living in that area. So we had to limit the study by that characteristic. However, they tend not to move, and we're concerned that the exposure to a specific genotype might be concentrated there. So we can ask the question, are the HPV genotypes different between women in the non-Appalachian area and the Appalachian area? We also wanted to ask about difference between race, so we'll do that in the non-Appalachian white women compared to the non-Appalachian African American women. And finally, a study that really hasn't been done before is asking the question about whether there's a difference in HPV genotypes between women diagnosed with CIN3 and those diagnosed with AIS. What's interesting about this is there are 48 labs spread across this 400 by 200 mile state. And we've put patients into the 4 categories that are necessary to answer those questions. We oversampled, so these are random samples of the population except for the AIS, in which we really only had 154 patients, so we used them all. Oversampled to 200, and in 5 months we were able to get 80% of all the samples needed for that study. It shows us that we can actually do this process and get a population-based result, and in 9 months we are almost up to 90%. A different study, and I'm going to do this really quickly using the same concept, has to do with, we have 4 investigators looking at 5 proteins: PAR4, snail, twist, Wnt, and ABLE and its homolog ARG. And all of these investigators have been NCI funded to understand the pathways that these particular proteins work in. They're all different pathways, they all do a bit different types of things. Some downregulate apoptosis, some promote cell proliferation, and so on. What we know about the recurrence of disease and the occurrence of disease is it's probably not one pathway, so it's hard to understand that in the lab alone. We built a retrospective cohort. Think about this. Of all the women, we have a registry, we have everybody, so we have all the women who had breast cancer. It was their first cancer, they had surgery, and they were determined to be disease-free following that surgery, and finally they could have been disease-free for a minimum of 5 years. And so retrospective cohort, we follow them forward and find that about 12% of them recurred, just about what you'd find in the literature. So then we went to all of those labs, got all of the tissue blocks, we anonymized them, used our research lab at the Cancer Center, built TMAs, stained for all 5 proteins, controlling for ER/PR and HER2, and we're now in the process of analyzing that data. So it's a really interesting way to be able to actually ask some very important questions. If we find a signal that more than one of those proteins are active in the set that recurred versus those who didn't, it drives the next set of basic science research. What's the link between these? How does it lead to carcinogenesis? And if we disrupt one of the pathways, is that sufficient to prevent carcinogenesis from taking place? Now I'm going to quickly go to this translational business. Used to be from the bench to the bedside. The more modern moniker is from the laboratory to the population. The idea is that basic science research can lead to human trials, which can lead to the last step of translational research, which is the broad-based implementation in the population. Example of this is one of the people on our cancer prevention and control program is looking at quercitrin, which is a naturally occurring byproduct of apple peels. It's been shown in animal studies to be very effective at preventing UV-induced tumors. If that could be made in the pipeline to a product that could be tested on humans, turned out to be efficacious, we could then do the broad-based implementation in the population to see what its effect was in the population. This just points out that it doesn't always start in the lab. Oftentimes, this will start with either an ecologic or a person-based study in the population, leading to some basic science studies, which can lead back to human studies, and then to that last step of the broad-based implementation in the population. I showed you the arsenic and chromium related to the high lung cancer rates. We also have in that same geography extraordinarily high rates of colorectal cancer. There is not a lot of literature on the relationship between arsenic and chromium as potential carcinogens for colon cancer. There is some work from Taiwan that suggests this. So we went and did some cell line studies and some animal studies. This is just a quick snapshot of the animal studies. We irritated the bowels in the second panel that you can think of as a control group. Then we irritated the bowel, exposed them to arsenic, and they all produced colon tumors. The next set of mice, we irritated the bowel, exposed them to chromium, they all expressed colon tumors. The last set, we irritated the bowel, gave them both arsenic and chromium, and we got these huge exponential tumors. So if we can find a way to disrupt that process that could be proven in human trials, then it would lead back to the broad-based implementation in the population. The point here is, regardless of where you start, it's this last step that we trip over most of the time. That broad-based implementation in the population has enormous impacts, and we really need to pay more attention to it. The ultimate goal in translational work is to take the efficacious findings from our large, usually phase 3 clinical trials, and make them broadly applied in the population. And it's not the goal of that last step, this is really important, to prove that that causes the change in the population. We did that in the clinical trial. The point here is to measure how broadly you implement it in the population. Hopefully you can discern from that who it's effective with and who it's not, and see if you get a change in the population. That is the goal of the last stage of translational research. So to do that, you really need to understand characteristics of your population. I'm going to talk a little bit about what it looks like in Kentucky, but I believe in conversations with people here that it's similar to what you find here in Alabama. Now, Appalachian Kentucky covers most of the eastern part of the state of Kentucky, but Appalachia is by no means a homogeneous area. So this is the catchment area, the primary catchment area of our Markey Cancer Center, particularly the part within the boundaries of Kentucky. But issues of poverty and literacy, as were mentioned by other presenters, have enormous impacts on what happens. This area, our catchment area, has extraordinarily high levels of poverty. More than 40% of the population living in many of those dark blue counties are living below the federal poverty guidelines. We have enormous issues of literacy. Literacy operationally defined as the proportion of the population over age 25 with a high school diploma. More than 70% of the population in some of these counties do not have a high school diploma. Those are significant barriers. Another measure is distressed counties, which combines those and other measures, and you'll see the concentration in the catchment area that we really want to make a difference in. It's important to understand those issues. And the issues of poverty and literacy have consequences. Top table is incidence, the bottom table is mortality. If you look at all cancers, the Appalachian area of Kentucky has extraordinarily high incidence and mortality rates for all cancers compared to both the state and the US. If you look at lung cancer, the state has the highest lung cancer incidence and mortality rate, but it's much, much higher in the Appalachian area for both incidence and mortality. In fact, it's some of the highest in the country. Same with colorectal cancer, and we also have very high rates of cervical cancer incidence and mortality. With breast cancer, our incidence rates are significantly lower, but our mortality rates are significantly higher. That almost always means, I know about overscreening the population. This is an example of underscreening the population. This is not adequately screening the population. There's a ton of literature to support this particular premise. So these four cancers, lung, breast, colorectal, and cervical cancer, have two characteristics. One, they represent 30%, 38% of all the cases that occur annually in the US. And the second is there are evidence-based interventions for each of these cancers. In Kentucky, they actually represent 42% of all the incident cases because of the high lung and colorectal cancer incidence. In the US, for mortality, these four cancers represent 45% of all cancer deaths. In Kentucky, it's over half. So if we do something with these four tumors, we will have an impact on the disease in the population. So this is our particular three-legged stool, if you will, for trying to move evidence-based interventions, both treatment and cancer prevention and control interventions, into the population to see if we can make a difference. The first is the Kentucky Cancer Consortium. This is a CDC-funded comprehensive cancer control program. It brings together more than 55 state agencies and organizations. Our key actors are the American Cancer Society, the Public Health Department, both of the universities, 2 major cancer centers, a large number of other agencies and organizations. And they do 2 things. One is they build a statewide plan, and this is data-driven, and the second is they have the ability to do policy initiatives. Fortunately, the American Cancer Society has public policy advocates who are also registered lobbyists, and that helps us. So the KCP is a really unique organization, and its job is the implementation of that state plan at a more local level. Now, Kentucky's a state with 120 counties. Some of those counties are very small. Robertson County has 2,000 people, and then Jefferson County has 750,000 people. So rates would be very unstable, and it wouldn't make sense. But what we've clustered together in area development districts over the years are people who belong together by the way they seek service. It can be buying goods and services, where they get health care, where they go to school, and so on. So those 15 area development districts each have district cancer councils. We have staff who have developed these cancer councils, so these are community-based folks. And yes, they include the usual suspects. Our local ACS people are there, local public health departments are there, local providers, both clinical and institutional. We have cancer patients, but what we have done is we have tried very hard to get individuals on those councils who represent low literate and low income populations. So the voices of the most vulnerable populations are present. And then the final leg of this stool is the Kentucky Cancer Registry, our population-based, statutorily driven surveillance program. Each year we develop an index of the burden of cancer by combining the registry data with other information. I'll talk about that in just a minute. We take it to the consortium to adjust the cancer plan, and we take it to each of the 15 area development district cancer councils, and it's their job to take the priority cancers in their area, and they do vary by area, and to implement evidence-based interventions, and then we go back each year. I know each year may be too soon, but it's good practice to see if we're making a difference in those priority cancers. So let's talk about the data that we put together. The demographic data include measures in the population in those 15 area development districts of literacy and poverty. And those actions of literacy and poverty influence risk behaviors like smoking or not being screened, which in turn have a direct influence on the incidence and or stage at diagnosis, and then contributing to mortality. So it's a pretty simple linear logic. The sources of the data are really nice because they're available in all states and most US territories. We use the census for the characteristics of poverty and literacy. We use the CDC's behavioral risk factor surveillance system. You may question that, but we have incredibly strong evidence that this seems to work in a population. We use the incidence data from the population-based registry and then mortality data from vital records. I don't want to go into this except for the headings, the column headings. If you see, we have the US, Kentucky, in each of those 15 area development districts. We operationalize the definition of literacy as the proportion of the population over age 25 with a high school diploma. We measure poverty by the proportion of the population living under the federal poverty guidelines for the 5-year period. Our risk factor for lung cancer is the prevalence rate of smoking from the BRFSS. We lag that by 5 years. It probably should be lagged by more, but this is just an intentional way to remind us that this is a cumulative effect. Then we have the incidence rates. Stage is not something we use because we, until very recently, have not had a way to find lung cancer early, and it will be a while before we can see the true impact of the broad-based implementation of spiral CT. And then we have mortality. So I'm going to show you some Pearson Productmoment Correlation coefficients. I just love to say that. I have no idea what it means, but it's always fun to say. No, actually I do. So if you look at the bottom axis, the percent of people with a high school degree, that represents the percent in each of those little blue diamonds. Those are each of the area development districts that have a high school education. And then on the y-axis, we have the percent in each of those blue diamonds that are living under the poverty level. And what you can see is as the educational rate goes up, the poverty rate goes down. And the square of that correlation coefficient tells us that 94% of that change in poverty is accounted for by the proportion of people with a high school degree. So that says they're highly correlated. So I'm just going to use one of those measures in the rest of this talk, and I'm going to use our operational definition of literacy. So if you look at another one, you'll see that the x-axis says the education increases, the rate of smoking decreases. And in fact, we're accounting for 82% of that decrease by that one variable, that literacy variable alone. That's a huge change. And we're accounting for that in a measure of smoking that has sampling error. So we sample telephone people for that. And it has ecologic error because we've clustered them together. And we're still accounting for 82%. That's really dramatic. Then as the smoking goes up, the incidence goes up, again, accounting for 70% of that change by the smoking. And then as the incidence goes up, the mortality goes up. These are highly correlated, approaching 90%, because the mortality rate for lung cancer is so high in that first year that you would expect this sort of correlation. Because these are correlated, we can build an index. I'm not trying to model mortality. I wouldn't want to do that because if you're a statistician, you would know that all I would get was multicollinearity. They're highly correlated. I've just demonstrated that. But I can use this to build an index using what's called a simple rank-sum technique. So in that first column labeled high school education, I've ranked them from 1 being the area development district with the very lowest proportion having a high school diploma to 13, the area development district with the 15, the area development district with the highest. 1 being the ad district with in the second column with the very highest smoking prevalence to 15 being the lowest. And then the third, 1 being the very highest incidence rate, 15 being the lowest, and 1 being the highest mortality rate in the last column to 15 being the lowest. I simply add up the ranks, and the ranks with the lower numbers are those with the greatest need for intervention. It's a pretty simple idea, and it's really easy to explain to people who may not have a background in statistics. So this is what we take to the ad districts. I just want to show you breast cancer really quickly because it's very similar, but there are some differences. We use education again, same way. Mammography is now the risk behavior, and it's the proportion of women over age 50 who've had a mammogram in the last 3 years, okay? And then because survival is so good with breast cancer now, about 10 years unadjusted for stage, we're really more concerned about the morbidity. So the stage at diagnosis becomes important to us. So let's look again at those correlation coefficients. As the education in each of those ads increases, the rate and proportion of women who've had a mammogram increases. In fact, 63% is accounted for by that one variable. If you look at many studies, we get excited if we get a 0.2, if we get 20%. We're looking at a huge relationship here that's very strong. And then, as the mammography increases, the incidence increases. This is really important. The more you screen the population, guess what? When you look for disease, what happens? You find it. So the incidence rate goes up. That's called the screening effect. It is not an epidemic in the population. It is a consequence of screening the population. And then as the mammography rate goes up, the proportion of patients diagnosed with late-stage disease goes down. So there is efficacious elements of screening. I know the overscreening argument, but I don't think we want to throw this out yet. So again, we do the same rank-sum technique, and you sum up the ranks with the 1s being those with the worst characteristics and the 15s being those with the best. What I want to point out is the incidence rate. We consider a low incidence rate a bad thing because it almost always represents underscreening. And look how correlated those are. It really is a good marker for that. So that's how we decide to intervene. I'm going to end this by talking real briefly about how we actually do this. And I'm going to use colorectal cancer as an example. Back in 2001, we noticed that Kentucky had the very highest colorectal cancer incidence rate, age adjusted to the US 2000 population. Compared to all other states, the District of Columbia was 63.5 per 100,000 population compared to only 54.7 in the US. We also found in that same year, looking back at the BRFSS, that we had the second to the lowest screening rate, 49th out of 50 states, having screened only a little over a third of the age-eligible population, people over 50, ever having had either a sigmoidoscopy or a colonoscopy. So we went through the process I just described. We took the data to the Consortium Committee, and they used their policy group and convinced the legislature to pass two bills. One required insurers to cover the cost of colonoscopy for age-eligible asymptomatic patients, and the second one provided funding and some educational resources for people who did not have insurance. This was prior to affordable health care. Then we took the data to each of the district cancer councils, and they all implemented, all 15 of them, evidence-based intervention programs. Now, by evidence-based, I mean we did that internal validity study, and there are two ways to do this. One is academic detailing. This is a study by Dr. Mark Dignan, where he went to primary care physicians and worked out systems so they would actually schedule their age-eligible asymptomatic patients to have a colonoscopy. And the second one was using lay health advisors. In the rural Appalachian areas of Kentucky, you need someone who speaks the language. Many of these people don't have a primary source of care. And so in order to reach them, we used lay health advisors. And so I want to show you what happened after we implemented that. We went from 49th, with just over a third of the population, to almost 2/3, raising us to 23rd in 7 years. No other state got close to that dramatic a change. So we're very proud of that. Now you'll notice those rates are flattening out because we're running into those really difficult, intractable populations by virtue of literacy, poverty, and access to health care. But if you're familiar with the work of Wendy Atkins, she did a huge randomized clinical trial in Europe, 125,000 people randomly allocated to FOBT, fecal occult blood test, versus sigmoidoscopy, not going the extra 40 centimeters. And that was published in May in 2011 in Lancet, if you want a reference for it. And what that study showed was if you screened every person in a population one time between the ages of 50 and 65, one time, with just sigmoidoscopy, you would reduce the incidence by 30% and the mortality by 40%. Because you're finding those adenomatous polyps and removing them before they become cancer. That's about 1 out of every 14 colonoscopies. And you're finding cancers earlier. That's about 1 out of every 1,000 colonoscopies. So what happened as a consequence of that? Before we intervened, beginning in early 2002, the rates were actually increasing in the population of Kentucky. But since then, they've declined dramatically. That's a 24% reduction in the incidence rate of colorectal cancer in the state. And then for mortality, the mortality rates did not decline in the first 2 years after we implemented our intervention. Anybody have a guess as to why that is? Yeah, the people dying were diagnosed before we started the intervention. And subsequent to that, the mortality rate has declined remarkably. It's a 28% reduction in mortality. Now, we think a 24% reduction in incidence, which translates into about 230 people every year in Kentucky who used to get colorectal cancer who don't anymore, and a 28% reduction in mortality is a significant public health success. And I want to say we know we're not done. We have that challenge of the last third of the population, and that will be the vista that we're really going to focus on now. But we think it provides real evidence that if we work together and broadly implement, do that last phase of translational research, it's going to have a real difference on people's lives. So with that, I will thank you for putting up with me.